I think Dr. Asim has joined. I have made him the co-host, and he'll be starting as soon as he's able to connect. Dr. Asim, are you there?
Yeah, can you hear me?
Yes, you are loud and clear.
Good afternoon, everyone.
Swati, just introduction, please.
Yes, sir. So welcome back, everyone. I hope you all had a pleasant break. We now begin our technical session for the day with the second lecture. I am delighted to welcome our guest of honor, Dr. Muhammad Asim from the University of Surrey, United Kingdom. Dr. Asim is an internationally recognized cancer molecular biologist and senior lecturer known for his groundbreaking work in prostate cancer therapeutics. His discoveries have directly contributed to FDA-approved treatments for metastatic prostate cancer. After completing his PhD in Germany and postdoctoral training in the USA, Dr. Asim went on to make pioneering contributions at the Cancer Research UK Cambridge University, including identifying new therapeutic targets and defining the synthetic lethal interaction between the androgen receptor and PARP pathway. He leads the androgen signaling lab and has received prestigious honors such as the Young Investigator Award and the MRC Accelerator Award, and continues to bridge fundamental science with clinical impact. We are truly honored to have him with us today. He will be speaking on topic the cancer conundrum. Over to you, sir.
Thank you very much. I hope I'm audible.
Yes, sir. Yes, sir, you are audible.
Okay, that's great. Sorry, I'm new to this Google Meet. Can you see my screen?
Yes, sir, it is visible.
And you can see it in full mode, right?
Yes, sir.
First of all, I have to apologize because the link I was provided was not working. So I spent about 10 minutes on that link. I am going to give you a brief overview of the cancer in this lecture. And if you feel like asking me any questions in the middle of this talk, please stop me and ask me any questions that you might have. Swati, thank you for your kind introduction.
Just a
Yeah.
pause here, Dr. Asim. We are restricting the questions that are typed in the message box so as to create linear.
I understand. That's perfectly fine with me.
So you can
The only thing is
message box.
I'm not able to see the messages in the chat box. So
It's in
let's
the right-hand corner. If you see the call out, if you click there, you see all the messages.
I'm not seeing any screen. I'm seeing one presenter screen. I'm seeing one full mode screen. I have three displays here.
Look
But
for
yeah,
call out, and they will be typing their questions there, and you can answer them as you wish.
I can read it out if I
thank
get
you. That
into
would be
things, I'll
superb.
read it.
That will be superb. So are you able to see my laser pointer now?
Yes, sir.
And can you confirm where I am circling it now?
DNA damage?
On cancer, right? On the word
Yes.
cancer.
Yes.
Okay, perfect. Thank you very much. So I'll keep it short. Thank you for your nice introduction, Swati. I'll tell very briefly. I am from originally from India. I left India more than 2 decades ago. Did a PhD in Liebig University in Germany on the very field that I'm going to talk to you about, which is cancer. Sorry, my voice is very croaky today. What we call it, super flu. That's what I've got these days. So I'm sorry for my voice. I did a postdoc in the US following my PhD, and I was a senior scientist in Cambridge. I'm still affiliated with the University of Cambridge. That's where I'm sitting today, working from home today in Cambridge. I run my lab, as Swati mentioned, on androgen signaling, primarily working in prostate cancer. So with regards to this talk today, I would suggest you have a look at this book if you're students. I appreciate 95 or 85% of you are the students, and I apologize at the beginning for those who are, I understand, are academics, faculties, who might find it super basic or maybe irrelevant. I'm really sorry for that. But over time, as we develop this course journey, you will start to appreciate that there are content which are quite complex, comprehensive, but also top level to understand cancer biology. So with this, I would suggest that if you want to study more detail about cancer, I have a copy of this book with me, which is a quite good book, Bob Weinberg, The Biology of Cancer. So please refer to it if you don't have a copy of it. This is an excellent book to develop understanding of cancer. So you might ask yourself how new or how old the cancer is. Dr. Aziz, do they have all the participants have the access to the mic or not?
No, I think.
Okay, that's fine. That's fine.
That's why we put it in the message box.
Okay, that's fine. No, no worries.
Or you can do a poll. If you want to do a poll, you can go there as well
Okay.
and do a poll.
Okay, no worries. That's fine. So you might wonder how old is cancer, and that is something that this paper that published in Mail Online print media suggests. They found a prostate cancer upon scanning through digital imaging a patient, a mummy actually, who died of prostate cancer more than 2000 years ago. So if we are saying that we have somehow invented the disease of cancer, sadly, this is not true. Cancer is not a new disease. It has always been there. So in terms of you developing as budding scientist, most of the students, what would define cancer is a continuous and controlled growth. So that is one of the hallmark features of cancer. Cancer cells don't want to stop growing. Usually you have patterns in the body. So the old cells die and their place is taken up by the new cells. And that's how the homeostasis is maintained. The continuity of life goes on. So what happens if you lose this balance is that these cells really, and that's what I think, because they become too many, they can't really stay in one part of, let's say, geographical location. For example, when human came into existence, most people think somewhere in Africa. They stayed there for some time, but as the competition for many things, among them nutrition, for example, grew, they started to move out. And that is exactly what happens in cancer. The cancer cells, when they are abnormally proliferating, competing for nutrition, competing for a space, they don't want to stay in one place. They actually move out of the primary organ where the cancer is formed, and that is what we call malignant tumors. I'm sorry about the spelling. I'm using a British spelling here, and this is American spelling without a U, but carries the same message. Unless they can invade to the nearby organs or spread through a specific process, which we will know in subsequent lectures, there is nothing like cancer. So the hallmark of cancer is that they're able to grow legs, and they can actually go from one part to other. And that is what poses a very significant clinical challenge for the doctors to treat cancer. Many cancers, they need a specific classification to identify what cell type the cancer is forming from, and that's how we dictate the treatment of this disease. Some common types of cancers are shown on your screen, as you can see. So for example, you can see one of the most common cancers in adult men and these statistics are throughout the world, is the cancer of the lung. In the both women, the percentage you can see in adult cancer is low, but then one of the most prominent cancers in women is the breast cancer. There are some statistics on childhood cancer as well. So where is the cancer forming majority of the time? And this is happening actually in the very cells that are at the forefront of dealing with external environment to keep us safe, to keep us maintain homeostasis. These cells are at the forefront. We call them epithelial cells. These cells give rise to full two-thirds of human cancers, which is a significant number given the epithelial cell is just one cell type in the body. There are many other cell types, as you know more than myself. I have specialist knowledge on prostate, breast, and some pancreatic cancer, but I don't know very much about other cell types. As you specialize, you become to laser focus on few things, and don't really appreciate other cell types. But this is quite important and remarkable that many of the cancers of human originate from epithelial cells. The cancer that Dr. Aziz laboratory in AMU studies, the cancer of colorectal cancer, actually the very cells, epithelial cells, become abnormal, giving rise to cancer. That's what he studies. I hope that's clear. Swati, if there is any question, please feel free to read it to me, and I'll be more than happy to answer them. If not, I will, for the sake of time, move forward.
Yes,
And okay, thank you. And you can see here in this electron micrograph something very prominent. These are the epithelial cells sitting on the basement membrane. And underneath the basement membrane, you see the connective tissue. This is by and large the normal, regular architecture of epithelial cells, be them on our screen, or these are the cells inside our organs. For example, the cells in our glandular ducts and so on. So what happens, one of the ways in which, if there is any pathologist, they can elaborate it more, but many times what we see as pathologists in the clinic using the biopsies which patients are asked to give is, at least in prostate cancer, whether the basement membrane is intact or not. The way the cancer cells are going to escape into the tissue, connective tissue, and then gain access to the wider body in order to spread, you have to break the basement membrane. There is a whole area of biology which is focused on proteases, which actually help melt down this basement membrane in order for these epithelial cells to gain access into the connective tissue. And that allows them to then spread. The scientific term that we use for the cancer, which is formed of the epithelial cells, we call them carcinomas. Anytime you see on any report, as you're developing into this field, cancer research, you might find people bring reports, and sometimes they say adenocarcinoma of lung, simply means lung cancer. And you can see here, as what I was alluding to earlier, a lot of human cancers are actually carcinomas, meaning they are formed of the product of the epithelial cells. You can see here on this bar chart on y-axis, there are cases, some statistical data from US, although the data is old, but the trend worryingly still continue. And I want you to focus on two terms that I've mentioned here, incidence and death rate. What you're seeing on x-axis, you're seeing different cancer types, and all of them are cancers, mostly of epithelial cells, leaving the leukemias and lymphomas. But have a look here, both of these, prostate and breast, they are the cancers which are the product of the epithelial cells. In green, you're seeing a lot of patients get prostate cancer, over 300,000. And this is, as I said, old data from the United States, just one country alone. And you can see some patients die of it. Recently, there was a recommendation by the government body in United Kingdom, which may restrict the screening to only cohorts which are more vulnerable, meaning if a first degree relative had prostate cancer or any other cancer type, those are the individuals who will take part in this screening program, which simply means taking the blood out and looking for a blood-based biomarker. I'm sorry I missed the earlier talk on biomarker. That was an interesting talk, but it is too early. I'm sure you can appreciate the sun just came out in UK. I could not attend that. So what happens is you can see that sometimes we might actually overdiagnose cancer. The reason clinicians are not happy about it, because a lot of times, especially in the United States, Dr. Aziz can tell you more about this. In the United States, a lot of time cancer surgeries are done even when they are not required, because there is no evidence that having a surgery done in instead of keeping a patient on a watchful waiting, meaning they have a regular scan after 1 year or 2 years, it actually makes no difference. The only difference is surgeries are much more expensive in US. I'm not saying imagings are cheap in US, but for example, the economies like United Kingdom, where everything is free in terms of medical facilities. When I go for any MRI, I don't pay a penny, a single rupee, I don't pay that. So that is important for economies like us to not overdiagnose cancer. And you can see here, there is a tremendous cost associated with this. In terms of Indian rupees, perhaps the government spends 5 crore on one patient cancer treatment over 1 year period. On the other side of a spectrum, you can see diseases like lung cancer. And here you can see incidence and mortality are almost similar. Or in pancreatic cancer, you can see as many patients as get pancreatic cancer, they actually unfortunately die of the disease. There are different reasons behind all these mortality or death rates. I'm not going to go into the details for the sake of time and that we're just starting kicking on this lecture series. But if you will be interested, please have my email address, and I can certainly take time out to discuss this in one of my subsequent upcoming presentations. So I hope this is clear up until now. Okay. So I mentioned that epithelial cells are the ones that get lots of cancers in them. What is causing them to develop cancer in them? The simple answer is they are the ones being exposed to something we call carcinogens. So, for example, when we go out in sun, our skin is exposed to ionizing radiation from sun rays coming from the sun in a specific parts of the world, and also given your skin color, cancer can form. There are lots of chemicals in cigarette smoke. I hope by the end of my lectures, I would be able to convince you to stop, if any one of you are smokers, to stop smoking. A lot of these cancers are caused by agents present in cigarette smoke. There are also not only these chemical and rays. I just want to appreciate the diversity of these agents. I'm not calling them chemicals because ionizing radiation is not a chemical, but given a different variety of them. For example, there are cancers known to happen as a result of viral infection. Typical example would be the liver cancer caused by the infection and not being treated for diseases like Hep B and Hep C viruses. And I think we can certainly classify hereditary predisposition and as one of the carcinogenic triggers, because if there is someone who has their first degree relative as being a cancer patient, the likelihood of having a positive cancer diagnosis in their progeny or their first degree relative is much higher. It does not look all very bad. You can see here, I'll show you in some next slides. You can see skin cancer is not very common in India, thanks to our skin color. Lots of melanocytes, the black pigment making cells stop us from getting exposed to cancer rays, harmful rays from sunlight that cause melanoma. 2 years ago, I lost one of my best colleagues in Cambridge, just my age. Turned out he had melanoma, which was metastatic. It spread to brain, liver, and despite being in one of the best places for cancer surgery, we have a cancer hospital, dedicated cancer hospital. And you might wonder, there are many hospitals. It's a cancer research hospital. So people do research on cancer and treat. So everyone who's treating cancer is actually a cancer researcher. Not that they once did some research on cancer and now for their living they only treat patients. No, they do active research. So this is one of the very common cancers in this part of the world, but also, as I said, the incidents are much higher in Australia, least in Japan. You can see cancers of nasopharynx, which is something which is giving me a trouble today to talk to you due to the infection, nasopharynx part, and the cancer of this are most common in Hong Kong, thanks to the air pollution in that country. Least in United Kingdom, if you go and check the air quality index of where I'm sitting, it is about 30 versus Hong Kong or some cities, industrial cities in China, where it is 300, 400 plus. So these are the cancers which are caused by these pollutants. You can see here prostate cancer. It's very common in US and least common in China. Part of the reason could be underreporting cancers in China. Not everyone will have excellent facilities to go and get themselves diagnosed if they have problem in passing urine, for example, and they feel their bladder is always full. Compared to US, where everything, if you have an insurance, you will not have much problem in getting yourself into a clinic. So there are different cancers, as you can see on this screen. Some of them are higher incidence, for example, pancreatic cancer in US, least in India, or lung cancer, most common in US, least in India. Okay. And you can see here that that's a good news I want you to look at. Have a look at what percentage of cancers are lifestyle caused. You can see here tobacco-related cancers, 34%. So 1/3 of the total number of cancers are caused by the tobacco related. So this might be an active smoker. It could be someone who eats penny, gutka, and so on. It could be someone who has a buddy who is a chain smoker. He himself does not smoke, but the buddy smokes. And likelihood of them getting cancer as a result of being a secondary smoker is much more higher. So please avoid having friendship with people who are chain smokers and do so in front of you, or discourage them at least not to smoke. You can see here there are diets which cause cancer. Dr. Aziz can tell you, he's an expert on colorectal cancer. Dye, which is less in fibers, very high nitrate, salt, can cause many cancers. And sadly, these cancers are very, very common in places like India, thanks to our food habits. So stomach cancer, esophageal cancer. I myself have a condition called Barrett's esophagus, and that predisposes me to developing a cancer of esophagus in future. So I'm always on, where is it called? I'm always on this omeprazole drug in front of me that hopefully will keep me safe from developing this cancer type. What you can also see is there are tobacco-related alcohol also causes lots of cancers. If you combine all of them, you would see that more than 70% of human cancers can be controlled by lifestyle changes, by adapting healthy lifestyle habits. So it's up to you. Many times people ask me, lots of reports are sent, especially from my home country, India, where you're sitting, most of you, by the way. Can you have a look at this patient's report? There are some obligations. Have a look. Most of the time, lung cancer or patient, they're smoking most of the time and developing cancer. So I'll show you some data on alcohol as a cause of cancer. Alcohol has been consumed by humans for millenniums, as you know. The fermentation was one of the earliest thing invented by humans. And you can see on this chart, there are at least 7 types of different cancers shown here. And the purple circle, you can see here, the bigger the circle, the higher are the incidence of cancer for that particular organ type. For example, bowel cancer, which is nothing but large intestine or colon, as we call it. Bowel cancer cases are the most common among drinkers. Sorry. Breast cancer also is very, very common in women who consume alcohol. I'm not saying males don't get breast cancer, they do, but the frequency of women getting breast cancer as a result of drinking is much substantially higher. And you might ask me, how does alcohol cause cancer? We have been discovering it every decade, a new thing that alcohol can do to the body, and I hope I'll have time today. I have 15 minutes. I'll try and finish this as soon as I can. But I'm not just regurgitating everything. I want you to learn this, especially as I mentioned, most of you are a young generation, Gen Z, want to learn more about cancer. If there is something you want to ask me, feel free to ask me. We will have sufficient discussion time. So back on alcohol, the way alcohol causes cancer is by damaging cells, and I'll come to it in a minute, what I mean by damaging cells. It can actually also exacerbate, it can increase the damage caused by tobacco smoking. So imagine someone having a cigarette, smoking a cigarette and drinking alcohol makes a very deadly combination, actually, to cause cancer. And in women, as I mentioned, lots of women get breast cancer. I understand many women in India are not smokers as much as they are in the Western countries, but primarily it can alter the consumption of alcohol, can alter the hormones, so the female hormones that we will learn in some of the subsequent lectures, can actually give rise to cancer. But also, importantly, in the last decade, we have discovered that there are compounds which are breakdown products of alcohol, which can actually give rise to breast cancer causing chemicals, which is quite alarming. So if you thought alcohol is bad enough, 7 different types of cancers, have a look at okay, I think that slide is slightly delayed. So I just mentioned to you the alcohol can give rise to breakdown products and they can cause cancer. Have a look at this paper published by the lab in Cambridge, Ashok Venkataraman, who has now moved to Singapore, I believe. And Ashok's lab did this research. So there are compounds formed in the body naturally, but their formation, their concentration is increased in patients who consume alcohol. And those of you who are very good in chemistry would know that alcohol, as a result of its degradation, will give rise to aldehydes. Well, sadly, aldehydes are the ones, it seems, behind the increased number of cases of breast cancer, because aldehydes can get rid of a good gene. And I'm sure many of you already know about what I mean by good genes. But these are the genes who keep us safe from developing cancer. I'll have some extensive discussion with you on this topic in subsequent lectures. But have a look. There is this good gene, BRCA2, which keeps us safe, both males and females, from developing breast cancer. And what happens, formaldehyde, in this paper published in Cell, Ashok Venkataraman's lab found that patients actually the consumption of alcohol and the formaldehyde it creates can actually degrade BRCA2, the good gene, by proteasome. So it is removing a good gene which is keeping us safe. Your guess is as good as mine. What happens if you don't have a good gene which keeps us safe from developing breast cancer? These patients will be much more likely to develop breast cancer going forward in their lifetimes. So you can see here the transient exposure. You can go and have a look at this paper, read it in more detail. If you can't access it, please let me know, and I can share this paper with you. And then recently, you can see this paper came out based on the research. This paper, the World Health Organization came up with this press release, and they mentioned about 3 years ago that no alcohol level is safe for human consumption. And they say very clearly in this that the risk starts from the very first drop. So when I was showing you that alcohol, how old is cancer, sorry, I showed you a mummy in which the cancer was shown to form, and that was more than 2,000 years old. Now you can correlate back then, 2,000 years, we have plenty of alcohol. We know how to make ferment glucose from alcohol. Those Egyptians were very fond of drinking, and I will not be surprised if they found out that mummy. Well, you should know that not everyone was mummified. Only the rich in those societies were mummified. They had very good access to, among other things, very good access to alcohol. So I'll not be surprised if that particular mummy died as a result of extensive consumption of alcohol. Sorry. So if you think alcohol is bad enough with 7 types of cancers, have a look here. 14 different types of cancers can be caused by the chewing of tobacco, which we do a lot of time. It's a very common thing in India, but also directly through the tobacco smoke caused by the cigarettes. And you can see one of the most prominent types of cancers is the cancer of lung. And you can see here, this is the data from Cancer Research UK. There are different kinds of cancers which are caused by tobacco smoke. And now you can follow this journey. Have a look here. What will happen here? Lung are being directly exposed. So anything in tobacco is causing that cancer. But you can see here mouth cancer, esophageal cancer, larynx cancer, pancreatic cancer. These are the number of cases which are caused every year alone in the UK. So if you ask me what is the population of UK, I would say it's 1/3 of the the Uttar Pradesh in India, which is one of the biggest provinces in India. So you do the maths and find out how many cases of lung cancer will happen in India with population more than 20 times the size of UK. So there are lots of cases suddenly caused by tobacco smoke. Dr. Aziz, are we doing good with time? When do you want me to stop?
Yes, I think you're fine. You have got time until 3. So we have got plenty of time. But that is a discussion as well.
Yeah, so is it okay if I go ahead for 10 more minutes?
Yes, absolutely.
Okay, thank you very much.
Sure.
Okay, so I spoke to you about viruses that cause cancer. Well, introduced to you these rays coming from the sun, harmful sunlight. But what about these chemicals that can cause cancer? And there is no functional chemical type which has been identified. Okay, these are the ones which cause cancer. They are actually quite different. They have a broad chemical structures. that can cause cancer, which we call them chemical carcinogens. Some of them are direct acting, others are indirect acting. Those of you who took modules like pharmacology would know what I'm talking about. I want to keep it assimilative for all of you, that all of you understand it. So I again apologize if someone of you is an M.Pharm, a scientist who has done Master of Pharmacy. They know very well what direct and indirect acting means, but I'll try to explain it in brief to give you the flavor of what I mean. And when I say broad range of chemical structure, have a look here. There are different chemical structures of these chemical compounds which can cause cancer. And this is benzopyrene, one of the favorite ones I learned as a student myself, found in cigarette smoke. That was actually one of the questions in the CSIR exam. So the question is, how do carcinogens, let them be viruses, let them be rays coming from sun, can cause cancer? Well, they cause cancer by causing mutations. I'm a scientist. Unlike most people sitting in US, I also teach students. Like we do in India, we run labs, but we also teach. And I think it's a very good thing to teach, give knowledge to students who you are discovering in your lab. It doesn't happen in America. You either teach most of the time, you're a teaching faculty, or you are a research faculty. In my job, it works most of the time 50-50%. So you're teaching. And so coming back to this, why am I telling you this? Because one of the favorite questions I have in the exam is, what define mutations, what they are. And trust me, 90% of the students don't know what mutation, how to define mutations. And it is very simple. It is a change in the sequence of DNA bases of DNA. How many bases am I talking about? 3 billion bases. That's the size of our genome, roughly. So these carcinogens can actually cause cancer by mutating, by changing the sequence of the DNA. And the system tries to suppress any changes. How? As you know, there are codons, same codon for different codons, sorry, for the same amino acid. But eventually, if you have any codon which is formed of a different amino acid, the protein is going to have a different amino acid, and that might alter the protein function. And how do we get this changes? Is it only caused by carcinogens? Well, most of the time, perhaps I would say yes. As I said, well, we have, people who are centenarians who have lived more than 100 years in parts of India, especially the northern parts of India, Pakistan. The average age is about 100 plus. And one of the reasons is very healthy diet, for example. But do they not get exposed to carcinogens? Well, they do, but probably much lesser than we do in our parts of the world. So when it comes to changes, the events that precede, events that are happening before this mutation, before the change happens in this DNA sequence, what is actually happening in the DNA? As you know, DNA is a very dynamic structure, but its dynamism is restricted to its hydrogen bonding. You might say, what I think DNA as the most stable structure, given the strength of the phosphodiester bond with which the two nucleotides are bound together. Those are one of the strongest bond. But as you would appreciate in the next lecture, the system, when it goes wrong, actually can give rise to these mutations. And why does it happen? Because the system is trying to correct any of these defects caused by these 3 things on the slide. So oxidative damage, for example, is the first thing. The second is the spontaneous purine hydrolysis. So oxidative damage, purine hydrolysis, and deamination. If you take out these 3 from the picture, if there is a way to make a human body which does not have oxidative damage, hydrolysis of purines, and deamination, cancer, to find a cancer patient, you will have to go to next city, if not not next state. So these things are so important in giving rise to a diagnosis of cancer. Sorry about that. So I will refresh your memory about how the nucleotides are formed, as you can see here. And why this is important, because I'll discuss you in brief the pathways which can actually cause the defects in these pathways can cause cancer, while trying to repair these defects: oxidative damage, the purine hydrolysis, and the deamination. So what happens in a nucleotide is that there is a pentose sugar made up of 5 carbon, you all know that. And then what is attached to it is a phosphate on one side, on this carbon, 5th carbon. And what you have on the other side is a base. The bond between a base and the sugar is a covalent bond, very strong bond. It's called glycosidic bond. On the other side, you have this ester bond. So those of you who are chemists or trying to study for CSR, you will know ester bond will be a functional group R, and then an oxygen and an R. This is called an ester, right? But if you place in between this a phosphate, it becomes phosphoester. Very good. But if you combine these two nucleotides through a phosphoester bond, there will be 2 phosphates on both sides, not shown on this screen. And that's why we call it phosphodiester bond, because there will be, there will be single phosphate, but 2 ester bonds. So that's why we call it phosphodiester. So there are 2 bonds which are very important for the integrity of this DNA. One of the most important one is this phosphodiester. And what you're seeing on your screen, nucleotide triphosphate, is none other than the unit for the synthesis of DNA. It's that brick that forms that wall we call the DNA. And you can see there are 2 types of purines, A's and G's. And you can see there are 3 types of pyrimidines, C's, T's, and U's. Uracil, as you know, is not found in DNA. Am I correct? You will see in a minute. I'll try to finish this ASAP in next 4 minutes. And then I'll open the floor, as Dr. Aziz said, for questions and answers. So have a look here. This is the way they base pair. This is a rather very easy bond, hydrogen bond to break compared to the phosphodiester bond, which is a covalent bond. So this is 10th class biology. A's bind with T's and G's bind with C's. And what happens when these change? Of course, the binding partner will change as well. And you can see here, there are some most common types of mutations or the conversions of purines into pyrimidines that will be classed as mutation. Yes. So what is happening here? Adenine, which is a purine, converting into guanine. That's called a transition. Or sometimes purines might become pyrimidines and vice versa. We call it transversion. These are all very deadly changes to occur in the DNA. So why are they happening? I'll have a case study and ask you to appreciate that. And that starts with this compound, benzopyrene, which I said is found in good quantities in cigarette smoke. Well, if you think that benzopyrene is the only thing in cigarette smoke which is a carcinogen, which causes cancer, that's not true. In fact, there are many, many more chemicals found only in cigarette smoke that cause cancer. Benzopyrene is one of the best studied one, and that's why I'm discussing this. It turns out, and I'll show you that slide, that when the lungs of patients are examined, not by microscope, but by sequencing, to look at different genes and what happens, what sort of mutations are present in these genes. It turns out that there is a good gene, like I was mentioning to you, BRCA is a good gene. There is another good gene that is called TP53, and those good genes, they have mutations. And you can see their function is compromised by benzopyrene. In the previous example I gave you from that Cell paper, I was mentioning The formaldehyde, which is one of the breakdown products of alcohol, can give rise to cancer by getting rid of another good gene, which was BRCA2. In this case, there is another good gene, TP53, and same thing is happening here. It can inactivate the gene. So the gene is there. If you do Western blot analysis, you will find, okay, the gene is still there, but there are mutations in it. As a result, it might be degraded. If it is not degraded, you would see it on a Western blot to measure the protein levels between a wild type patient who never smoked, for example, and someone who smoked. Uh, so we will catch up on this in the next lecture. I am happy to stop and take questions, Dr. Aziz.
All right, so we have got plenty of time for some questions. 10 minutes.
Yes, more than happy to. Is it possible to open the floor so students can talk directly, Dr. Aziz?
Okay, we can do that. Or they can type whatever is
Yeah,
convenient.
they can, whatever you like, your choice. You are the chair.
Dr. Azal, can you just open the mics for everybody, please?
Looks like no one has any question. Either I was very clear or I managed to confuse every one of you. Sorry about that then.
I think everything was very clear and questions. This is such after the lunch session, so probably I on.
Go on, please.
Yes, hello, good afternoon, sir.
Very good afternoon. Go on, please.
Yeah, you have been very particular about everything. I just have
Thank
one
you.
question. Like, I have been in a
Sure.
dilemma that
Uh-huh.
whenever we study about whenever we study bioprocessing, we have been told that beer consumption is good for health, obviously in moderate consumption. But on the other hand, as we study, alcohol is carcinogenic. It
That's
contains
right.
oxidative stress and all kind of mutation it can occur, like gene issues can be done. So,
That's right.
what exactly can be said that beer consumption is it good in moderate consumption or not?
Well, as you can work out, I'm a Muslim by religion. I have never drunk any alcohol, and I never told my student before that study came uh, from Ashok Venkataraman's lab, and also by the recommendations by World Health Organization that the damage to our body starts with the first drop. So
Yes.
before that, I never said to students, but just because of my personal belief, I don't want to impose it on my students. But now I, would go and show them these slide and tell them, look, it's not really good. We have scientific evidence. So Aparna, for us as scientists, religion aside, we are meant to believe the evidence, and the evidence suggests that actually you can cause extensive damage to our body. And as you would appreciate in the next lectures, as we age, our system that deals with DNA damage actually becomes slowly and slowly weakened, and that's why the maximum damage can occur. So if I tell you the answer that the question, try to answer the question, I would suggest when you are very young, you might not see as many side effects straight away as you might see as growing old in terms of developing cancer caused by beer consumption. But looking at the evidence that our own body produces a lot of formaldehyde as a in a regular way as a part of our metabolism, what we are going to do is to overburden by that system by drinking more alcohol. And if some people, and I'm asked this question again and again, well, that person never smoked, they still developed lung cancer, why? I'll show you some some slides in the subsequent lectures. To tell you the truth, what I think is that there will be damage. I understand there is much lesser quantity of alcohol in beer compared to something like tequila or vodka, for example, but they all cause damage, certainly. And as you mentioned rightly about the oxidative damage is actually not a bad thing. Sometimes we need oxidative damage. And I'll give you some examples during this talk. If you're wondering why it is good, because it is being played as a black sheep, oxidative damage is bad. It's actually not.
Oh, okay.
I can tell
thank you
you
so
in
much,
more
sir.
detail. No
Yeah.
problem, Aparna. Thank you.
Additionally, I have one more question, sir.
Sure.
Similar question, like in foreign countries, like in UK or in more foreign countries, there is a
Mhm.
consumption of wine because the region is colder. So
Uh,
same thing.
yeah.
So there will be the same, like it is a silent killer for them too.
Aparna, if you come to hospital, so I was ill to the point that I wanted to see my doctor 2 days ago. I could not speak at all. And would you believe I had to wait 48 hours to get access to the simplest of the doctor we call the Mohalla clinic type of doctor. There are no doctors here. Why? Because most of them are busy treating patients who have been chronically ill. So you have a good medical system. You keep people alive for a very old age, 80, 90, 100, very common in this country. But at what cost? They are debilitated. They can't walk by themselves. They're ill. They have cancer. You're keeping them alive. You allow them to smoke cigarettes. I was speaking to someone who is a politician about 10 years ago, and I asked them, you can stop people. You make a law that weeds, the simple marijuana. You can't really smoke in this country. It's a law. You can do the same for cigarette. And the answer I got? Well, we spent 3 billion pounds on cancer treatment. Fine, but the revenues from the sale of cigarette is about 6 or 8 billion pounds. So despite that, so this is a human greed, perfect example of human greed, why governments cannot actually impose a ban, knowing based on very strong scientific evidence that these things actually are causing people. Cancer is the last thing, Aparna, you would think to happen to someone who's smoking or consuming alcohol. There are lots of things which we now know happen: ability to make judgments, ability to carry out regular works, mental health illnesses. Many of them have their roots in cigarette smoke or alcohol consumption. So we are actually same thing here. A lot of people drink, and drinking is much more common in this country compared to India, I would say. And therefore, you see lots of these effects in population. We are sick population.
Okay, thank
I
you so much
think
for your answers.
there
Thank you.
is a question
Yeah,
from
please.
Abdullah.
Yes, Abdullah, go on, please.
I can read it out for you. It says,
Yeah.
I have a question, sir. Why the statistics data about the prostate and lung cancer is more prone than any other part of cancer in the body?
More prone. Abdullah, static data is more prone. You mean more cases, Abdullah? If that is what you mean, there are more cases of these. There are two things which are happening in this case. Prostate, for example, happens to people who live long enough. Breast cancer is also very common. You're asking why they're happening a lot. One of the reasons is overdiagnosis. We are diagnosing cancer when there is something, Abdullah, called, and all of you perhaps know, it's called incidental finding of cancer, meaning some people in this country donate their body to hospitals, to medical students. Turns out they open the body, they use, of course, it very wisely. Not many people donate their bodies. They open them, open the prostate, do the histological analysis, and found actually there was a cancer in this prostate, which was never discovered, never caused any trouble to that patient, meaning that was an incidental finding that happened from an autopsy. So what we do is there are very easy tests to detect these cancers. How? Prostate can be detected, at least the initial diagnosis can be made by a blood test, simple blood test called PSA test. And that is rolled out to the entire population, 60, 70 million people in this country, meaning anyone can have their PSA tested. I'm nearing 50 years of age, and they have already started measuring my PSA every year. I go to the clinic for regular checkup. Of course, if you're going to do that, there will be overdiagnosis. Is the cancer only thing that increases levels of PSA? No, but it is one of the things. So if you overdiagnose people, in some cases, for example, that autopsy, the cancer didn't say anything to that person, but that person had a cancer. Had they gone when they were alive to a clinic, they would have a positive diagnosis of cancer, which did not happen because it never caused any problem. We don't go to a doctor unless we have any issues. So I would say many times it is the overdiagnosis of these cancers. Breast cancer, for example, 40 years of age, you go and get your mammogram done, ultrasound done. It's very common. Why? Because you would know 50% of the women in the world have something called breast fibroadenomas. These are small lumps in the breast which would never form cancer, never ever. But because 50% women have it, they will be seen, and many of them may have um, diagnosis of cancer. I'm not saying they should stop going to the doctor. I'm saying that we need to question ourselves: are we overdiagnosing those cancers? So you are right about that. Um, any Does that answer your question, Abdullah?
Yes, sir.
Thank you. Any other?
Yes, sir. We have another
I'm
question.
seeing. I can see that now. So the next. So Sandeepan is asking, alcohol consumption is indeed risk factor for cancer, but exposure like, sorry, it's moving quite fast. Exposure like using ethanol in the labs or simple hand sanitizer poses the same risk. Sandeepan, you've asked a very good question. I never thought about this. Uh, the thing is, I think we should do some studies to see when we are wearing gloves, is it penetrating through our skin? But I would say I would not worry too much on top of this, because we are not consuming it directly through our liver system, because many times the products that alcohol creates is through our liver system. So when it goes through the hepatocytes in our liver sometimes can convert things which are not really cancer causing into cancer causing things. Remember that direct and indirect carcinogen I was talking to you about? So sometimes there may not be chemicals which are causing cancer, but our liver can indeliberately convert them into things that can cause cancer. So you're absolutely right. That's a very nice question, very out of box thinking. But I would say I would not worry too much, because it's not passing through our primary, the metabolic pathways, not through being processed through our metabolism. So if it goes through the fingers or something, I don't think that will cause much damage. Then I have another question from Madiha. She's asking, sir, why some medicines such as cough syrups contain alcohol and are prescribed by doctors when it is known that it is carcinogenic? Again, excellent question, Madiha. And I think,